Hey guys, so last week and this week I've had you looking at viruses, and the reason I decided to do this is simply because, the whole situation we're in right now, specifically with that one virus, COVID-19. And so, normally I kind of just do a brief overview in this chapter, but considering our circumstances, I thought it would be better for us to take a deeper look than we normally do at these viruses. Maybe you guys have questions about COVID 19. Why is it so dangerous? Is it really that dangerous? What's happening? Things like that. And so that's kind of what I'm hoping to get started with you guys this week is just looking at that. We're not going to be talking much about COVID necessarily this week, but we are going to be looking at viruses in general, and a lot of this information will apply to COVID 19. And so, to even understand what viruses are, we kind of have to go back to the whole idea of germs and like what germs are. And germs is a very broad term, but nonetheless, we got to go back about 200 years to Louis Pasteur and his development of germ theory. So back in the 1860s, this is about the same time that we were fighting our civil war here. Over in France, Louis Pasteur was doing all these different experiments to prove that there was these microscopic organisms that were causing people to get sick. We knew about microscopic organisms like bacteria and protists, and we knew about them because we've had microscopes at this point. You can even see right here in this picture, he's got one right there to check his samples to see if he did or did not get bacteria in these flasks. And what he found out was that he wasn't getting it. But nonetheless, what this did was this helped him develop this idea known as germ theory. And it seems kind of weird to us today, especially in our modern situation with this COVID-19. We're always like, wash your hands, wash your hands, wash your hands, do hand sanitizer, this, that, keep social distancing. But before the development of germ theory, it was common for doctors to do one surgery and maybe they just amputate some guy's leg, but now they're going to go deliver a baby without washing their hands or doing anything because they just didn't think. They didn't know about all these microscopic organisms that were on them. In fact, until about this time, there was something known as, I believe, it was iasma, and this isn't asthma that you get, but the iasma was this idea that it was dirty air that got us sick. Nobody really understood what germs were. And so it wasn't until the invention of the microscope when we started being able to look at these things under the microscope and seeing that there were these microscopic organisms that we started to realize maybe they play a role in our life. And Louis Pasteur and others came to this agreement that it was these things like protists, these bacteria, all these microscopic organisms that were making us sick. And together they just grouped all these things, whether it was a protist, whether it was a bacteria, whether it was just some unknown microorganism, they called them germs. And so that's why you have the idea of a germ in your head when we talk about viruses or even bacteria or even just getting sick. You have germ X to get rid of these things. And so the idea of germs is just a broad way to say pretty much anything that causes us to get sick, any little microorganism. However, as we started to find out, these viruses don't even fit the category of a microorganism. And so let's take a look at what that means. So moving forward about 30 odd years, we get to Dmitry Ivanovsky, who was a Russian botanist. And what he was studying was the tobacco plant. And on this tobacco plant, he was studying this infection that it would get, which today we call tobacco mosaic virus. And what it does is it gives the leaves of the tobacco plant this mosaic patterning. And he believed that there might have been some bacteria that was involved in this getting the plant sick. And so he developed this plan to try and figure out how to see if that's what was causing it. In other words, what he would do was he would take one of these sick plants and he would grind it up or get like a bunch of the sap and he'd grind it up and then he'd put it through this filter. And at the time of him doing his experiments, these were the first of their kind type filters. They were so precise that they would even filter out bacteria, and he knew that. And so he figured that if I can filter out the bacteria from this sap and then take this sap and put it on another plant, like I'm trying to infect it, it shouldn't get sick. However, to his kind of surprise, he realized that even though, so he took the stuff from the sick plant, he put it in his filter, and he got this stuff that was bacteria free. Mind you, there is no bacteria here at all. And he put it on this plant, on the healthy plant. And he would expect that this plant would stay healthy. However, what he started to see was going on was that the plant was not staying healthy, but it was getting sick just like the other one. And so he realized that it's not the bacteria that's making it sick, so there must be something else. Not only that, though, this must be something that's small enough that it can get through that filter. And so he believed that what was happening was that this bacteria that was living on the plant must have been creating this toxin, this chemical. And it was this chemical that was able to leach through and poison the plant. And in fact, that's where our word virus comes from. The Latin root means poison. And so he dubbed it virus. And that's also why he's kind of considered one of the founders of virology, which is the study of viruses. That's kind of something important you might want to know. So like when we're talking about all these doctors who are dealing with COVID right now, they would be what we'd consider a virologist, somebody who studies these viruses. And so he was, because of this, and the idea that he came up with, saying that it wasn't the bacteria, but it was a chemical from the bacteria that was poisoning the plant, he's considered one of the first to discover these viruses. However, he didn't think it was a virus. What he thought it was just some chemical, which, again, a lot of people did, even up to the 1950s. People didn't really understand what viruses were, or specifically this type of virus. They just thought it was a poisonous chemical because they didn't see any biological reactions like you would see with bacteria when they extracted it, and so they just figured it was this non-living thing. However, we started to look at it, and this right here is the tobacco mosaic virus. We can see that it has some RNA, and it has this protein coat around it, and it doesn't really meet the criteria of being a cell, but nonetheless, this is so small that this is what was able to get through his filter. This is what was able to get through and was able to make the plant sick again. It actually had nothing to do with the bacteria. And looking at this now, let's go ahead and start to look at what it means for a virus to have this protein coat or this RNA strand here. And so we're going to look at the virus's structure. So viruses basically have two main parts. They're going to have a nucleic core, and when I say nucleic, I'm talking about a nucleic acid. So it's either going to be DNA, which stands for your deoxyribonucleic acid, or it's going to be RNA, which is simply a ribonucleic acid. And that's going to be just like it is in us, it's a blueprint for building proteins, so it's a blueprint for building them. And then to protect that information, they're generally going to be covered in a protein coat that's known as a capsid. Sometimes this capsid can be covered with a bunch of lipids, which is like the case with influenza here. It has this lipid envelope to protect it. Now, this lipid envelope is also the reason why washing your hands with soap is one of the best things that you can do to prevent the flu and even coronavirus. Because what's happening here is this lipid coat, what our soaps do, soaps are meant to pull apart lipids, and so the soap literally rips this apart, exposing the virus's capsid, and then exposing it to the rest of it, basically destroying the virus. And that's why they say soap is so important, because it destroys that lipid envelope. But nonetheless, your two basic components are going to be the nuclear core and the protein cover. Now, again, what you see here, another way that we classify these viruses aren't just by their DNA or their protein coat, but it's how that protein coat is structured. So we have like this just general tube-shaped virus here that would be the tobacco one that we were talking about. We have our influenza, which is this almost spherical one that has all these little projections or protrusions on the end. And in fact, looking back at our corona virus right here, these little things, corona means crown in Latin, so that's what we're talking about. That's another way that we classify them. But this guy right here, this guy you've probably seen before, and this isn't necessarily going to infect any of your cells. However, it may infect some of the cells living inside of you. This is what's called a bacteriophage. And these bacteriophages are sometimes used by doctors, but also in general, these are what are going to get in and they're going to wreak havoc on bacteria and different things like that. Our cells are going to be affected by these guys. Some plant cells might be affected by these, but bacteria are going to be affected by bacterial phages. And how they infect, how in fact all these viruses infect, it's going to be one of two ways. It's either going to be through what's known as the lytic cycle or the lysogenic cycle. We're going to do a brief overview here, but if you haven't watched it yet, I highly suggest going ahead and watching that Amoeba Sisters video on the viruses, because they do an excellent job on explaining these as well. And so if you want to do that, go ahead, head over there after you finish this video, but please do that. Alright? So the first up, before we even get to either of these, it's probably important that we go over something that we talked about last semester. Protein synthesis. Alright? Why is this going to be important? Well, because basically this is how viruses are being made. This is how coronavirus is being made inside of the bodies of infected people. This is how the flu is made inside the bodies of infected people. This is how bacteriophages are made inside the cells of bacteria. It all has to do with protein synthesis. So first of all, we know that our DNA, that's what stores our genetic information. It gets coded into RNA. That RNA is the only thing that can be read outside of the nucleus. Once that RNA is out here, what's going to happen is these ribosomes, alright, remember the ribosomes? Those little organelles that make proteins? Those ribosomes are going to read this message, and they're going to start to add these amino acids into this peptide chain. As they do that, it builds a protein. Sometimes those proteins have certain shapes like hemoglobin or hormones, and other times they're going to build proteins that look like this. That's the protein shell of a virus. And so, again, what's going on in these different cycles is these viruses are basically going to hijack the cell's ribosomes and use them to replicate themselves instead. And so what happens in this process known as the lytic cycle is we have our virus. This one again is talking about the bacterial phage, but this is the same whether we're talking about the bacterial phage or influenza, any of these. It's going to be using either one of these two processes that we're about to talk about. So in the lytic cycle, what we see happening is our bacterial phage, it's going to line up with the cell. And generally, sometimes some of these phages or other cells like influenza, they have those certain markers on the outside. And those markers can only line up with a specific cell. And so once it lines up with that, that cell is going to allow it to release its either RNA or DNA. And in this case, we have viral DNA. So that DNA is going to enter the cell, And once that DNA is in here, that uh, our ribosomes and everything are going to come together, and they're going to start to read that, and they're going to start to replicate it. Just like they would be replicating this DNA, now they're going to start replicating this virus DNA. And as they replicate this virus DNA, then those ribosomes are going to come along, and those ribosomes are going to start to read that DNA, and they're going to start to code to make the bacteriophage protein coat, this capsid. And as they make these capsids, and as the DNA gets moved into these capsids, it continues doing this over and over and over again until until the cell is so packed with these viruses that lysis occurs, this breaking down of the cell. That's why this is the lytic cycle. And so, long story short, what happens in the lytic cycle? The DNA comes into the cell. The cell then starts to read and replicate that DNA. As that DNA is replicated, it's going to start to read the DNA to make these capsids. And as you get more and more, and as they start to develop, then they're going to basically overpopulate the cell to where it explodes, thus killing the cell. This happens with the flu, when our immune system tries to fight it, or other types of cells inside of our body get infected. This is what's happening, is this virus starts to get replicated over and over and over again until the cell just can't take it anymore and it explodes. And as you have this going on at an exponential rate where it's happening over and over again, at each cell side by side over and over again, you all of a sudden start to see how you have this huge spread of virus. And once you get so many cells that are damaged, that can really weaken the immune system and the body in general. And so it's not a good thing. Up next, you have the lysogenic cycle. Just like our lytic cycle, what we're going to have happen here is our virus is going to find the cell, the target cell that matches it. It's going to release either its DNA or its RNA. And what's different about this one, this is sometimes what we'd consider almost like a retrovirus, because it's actually going to accept its DNA and it's going to add it to its own chromosome. Now again, this is a little bit different type of chromosome. I guess you just consider it the genome of this bacteria here. But nonetheless, this DNA from this virus is now here, and it's here to stay forever. Alright? Notice we haven't made any of the virus yet. It's just taken its DNA and it's incorporated into the cell. So the virus's DNA is this green DNA. The cell's normal DNA is this purple DNA. Now look what happens when the cell divides through mitosis. Alright, we had our green DNA again here. We have the green DNA here again. What happens is, now that this is here, it's here forever. And so when this cell gets ready to replicate, when it gets ready to go through mitosis and it reads this DNA to duplicate it, it's now going to duplicate this DNA. That's going to happen each and every time this cell replicates, over and over and over again. And sometimes it could stay there dormant for years until something happens where it actually gets under stress, whether it be like you're sick, or whether it be just heat, or even just emotional stress can cause these viruses to all of a sudden activate. One of these is, or not one of them, but one of the families of viruses that's lysogenic are the herpes viruses. One of those in particular, the herpes 1 virus, which is just your cold sores. So if you have a cold sore or a fever blister, that is a lysogenic virus. And if you have one, and then it goes away, you still have the virus in your system. It's just not activated. But let's say you have a lot of work coming up. You have a lot of tests, you have a lot of projects, it's just very overwhelming. And all of a sudden you start to realize your lip starts to hurt a little bit, and you start to get this cold sore that pops out. Guess what? It's because of all that undue stress that now this DNA that was dormant is going to start to have the ribosomes read it to replicate it, and you're going to have the same outcome as you did with the lytic cycle. And so again, this only happens though when the virus gets stressed out. So another example of this is chickenpox. Chickenpox is a type of herpes virus as well. And generally, people who are younger get chickenpox, but that doesn't mean you can't get it when you're older. And most likely, if you had chickenpox when you were younger, you will have the same virus pop up again. However, when you get older, we don't call it chickenpox anymore because now it shows itself as a new disease, shingles, which can be extremely painful. So, long story short, with chickenpox and shingles, chickenpox, you get the virus, and the virus is all throughout your body, and you get these sores and lesions all throughout the body. However, you get over it, but the virus is still there. And then all of a sudden you're stressed out, or your immune system is really weak, and you've just been exerting yourself a lot, and your body's tired. Maybe that's just the right time that the virus has been waiting for, and now it will reactivate itself, but this time is shingles. And shingles is really weird because it only affects one half of your body, and it literally, there's like a line that goes down from your back, and it's either going to affect your right half or your left half. But that's the shingles virus. It's the same thing as the chickenpox virus. So here's the other dilemma. Let's say you have a grandparent who gets shingles, because generally it's the older population that gets shingles. So let's just say you have a grandparent that gets shingles, and they have shingles. Let's just say it's on their neck. All right. You go in to give them a little side hug goodbye, and the next day you wake up and you start to itch. You don't have shingles, but now you have chickenpox because you never had the chickenpox virus when you were younger. Now let's say you did have chickenpox when you were younger. If you had chickenpox when you were younger and you come into contact with shingles, then you'll get shingles. But if you've never had chickenpox and you come back into contact with shingles, then you'll get the chickenpox virus because they're one and the same. The only difference is that because it's a lysogenic, remember, it's storing its DNA and it's not doing anything with it. Here's chickenpox, but then all of a sudden it calms back down. That chickenpox virus is still there until it's ready to become shingles. So that's what's going on with chickenpox and shingles. That's another great example besides just the other herpes one that we generally talk about. HIV, AIDS, that's also a lysogenic cycle. Now, are there ways to combat this? Well, in all honesty, once you have it, no, not really. However, there are preventative measures that we can take to make sure that when these infections do happen, or when these infections are prevalent, we're not going to be in as much danger as we would before. And that is with vaccines. And so, again, you guys should have read the other day in your textbooks, page 237, where it was that little section, Facets of Biology, that was talking about vaccines, and Edward Jenner, who was a physician in the late 1700s, and what he was looking at was the disease of smallpox. Now, this is kind of a hard example for us to kind of wrap our heads around today, simply because of how well his work actually worked with vaccines. Because of vaccines, we've been almost just about able to eradicate smallpox. But nonetheless, what was happening was smallpox was this extremely deadly disease that would result with these giant pus sores and lesions all over the skin, and it would get bad enough that you could even die from it. And in a lot of cases, a lot of people did die from it. Again, if you guys haven't, I would highly suggest watching the vaccine video from TED Ed that I sent you guys. It does a great job of explaining this, probably a lot better than I could. So again, go ahead and watch that as well. But it just kind of also lays the groundwork for about why this was so dangerous, because so many people died from smallpox. In fact, smallpox was one of the leading viruses that most likely wiped out a vast majority of the Native American population. And so it was a dangerous disease all throughout the world. However, there was another virus that was very similar to smallpox known as cowpox. And it's actually, we know today, they're kind of in the same family of viruses, but it's um its types of what's the word I'm looking for? Its infection wasn't as severe as smallpox. In other words, people who would get cowpox would recover from smallpox. And Edward Jenner noticed this. He also noticed that when these people who had gotten cowpox recovered from it, they were generally less susceptible to get smallpox. And in fact, they usually didn't get smallpox because they had had cowpox. And so this led him to this idea that maybe if people had cowpox, which had a lot better likelihood of survival, if I give them this disease that they'll get sick, but they won't get deadly sick, and then it protects them from the disease that will make them sick, maybe that's a way of protecting ourselves from smallpox. And so he went to all these milkmaids and everybody else who had the cowpox and identified different things, and then he started trying experiments. And one of them was on this poor kid. And long story short, what happened was his mother feared that he might have smallpox, and so she brought him to the doctor, and the doctor saw the boy and thought, this is the perfect case I've been looking for. And so he decides, all right, we're going to try this vaccine thing on your son. And so what he actually does is he made two small cuts into the boy's skin. And what he did next was he took like this powder and this pus from the lesions on somebody's skin that had cowpox, and he rubbed it into those cuts. So yes, in other words, the very first smallpox vaccine, or one of the first smallpox vaccines, was to simply make a cut in the skin, take some pus from a cowpox wound, and put it in there. And for whatever reason, he kind of hoped, maybe that would work. And so, of course, he had to know for sure. And so, a few months later, he called the boy and his mother back. He did those same two cuts, but instead of putting cowpox this time, he tried to give the boy smallpox. Remember, smallpox was the deadly one. He looked to see if there was any change. This time, the boy didn't even get sick. He got, I believe he got like a minor sickness from the cowpox, but he recovered. But when he got smallpox this time, there was nothing, no symptoms at all. He was what you've been hearing in the news a lot, asymptomatic. And so he left it for a few months, and then he said, I'm going to try it one more time. So he did it again. He did the cuts, he put the smallpox wounds, the nasty stuff from the smallpox wounds in those cuts, and nothing. And this helped him make the foundation for what he called these vaccines. And just like you have your anti-vaxxers today, it was even more prevalent back then because the science really wasn't known about it. He just knew that there was this similar disease. Again, they didn't even realize what a virus was at this time. He just knew that there was this similar disease, and if people had that weaker disease first, somehow their bodies would build immunity to the stronger disease. And that right there is the whole point behind vaccines. The point of a vaccine is to give our bodies generally a weaker strain of something so our bodies can build up what we now call antibodies to fight against it the next time it sees something like that or that same invader. And so we're going to be talking about vaccines a little bit more in the future, uh, but let's just do a quick recap of what we talked about today. So today we talked about virology, what it is, the study of viruses. We talked about how they were basically unknown until the late 1800s. Up until then, people didn't even understand what germ theory was, that there was microscopic organisms that could make you and other people sick, and washing your hands is a really important thing. And then we talked about when we first finally started to look at these viruses under our microscopes. And again, these viruses are so tiny that we needed really good microscopes to see it, like electron scanning microscopes, where it can see down almost at the atomic level. And when we finally started to look at them and take them apart, we could see that they were uh, either going to be a DNA based or an RNA based. And with that RNA base, they're going to have some type of protein coat. We talked about the two different life cycles of viruses, how they had the lytic life cycle, which simply is the virus invades, it repopulates, destroys the cell, or the lysogenic, where it invades, it stays dormant or latent, and then later on it will cause havoc. And then, so again, the lytic virus reproduces rapidly, destroying the host. The lysogenic virus can remain latent, which just means dormant, for a long, long time. And then last but not least, we talked about vaccines and how vaccines are important because they help our bodies build immunity towards viruses. Remember the cowpox, it wasn't as strong as the smallpox, but because we were able to survive that, our body was more prepared for when it came into contact with smallpox. It's the same reason you get a flu shot. Sometimes it's chopped up pieces of the flu that look similar. Sometimes it's a weaker strain of the flu. And that's another problem that we're dealing with right now with this coronavirus, is it's a new virus that our body has never been exposed to. And so we don't have any antibodies built up. That's why one of the newer tests that's coming out, the antibody test, is so important, is because it tells doctors if you've had the virus. And if they see those antibodies there, they know you've had the virus, and those antibodies might be useful and for making vaccines for COVID to come. So that's kind of just a basic overview of viruses in a nutshell. Tomorrow, you guys are going to be looking at a little bit more virology. We'll be discussing it. Next week, we'll take a little bit deeper look at COVID-19 specifically. And that's about it for right now. So if you got any questions, let me know on Remind, and I will talk to you later.